Okay, welcome to today's lecture, which focuses on the principles of disease, epidemiology, and diagnostics, which includes some various forms of biotechnology. Now, today's lesson is one of those lessons that students find especially interesting. Well, I think all of the lessons are interesting, but I'm kind of biased. And so the reason why students tend to like this lesson is it's very applicable to what you do every day, and it's very applicable to any of you working in the clinical area or the medical field. Now, I know there'll be a lot of terminology, which, sometimes you may kind of get bogged down with all of the words or find that a little boring, but again, this is one of those lectures where you can really see and understand the things that we're talking about, because they're things that are very visual to what you've experienced your whole life. So whether you've worked in a hospital or medical field, or you yourself having been sick, or your loved ones as well. So like I said, today there is a lot of terminology. And so the first bit of terminology in the field of diseases that we need to kind of distinguish amongst includes the terms pathology, etiology, and pathogenesis. And these each have their kind of distinguishing factors about what they truly mean. The first one is pathology, and as you can see from the beginning of the word patho, this one is dealing with pathogens. Specifically, pathology means the study of disease. The second word here, etiology, now we're looking at the cause of a disease. So for instance, trying to identify the exact bacteria that causes a particular infection. The next term is pathogenesis. Now, patho you saw as disease in the first term, but now when you include genesis, anything that involves genesis talks about the beginning of things. So pathogenesis is defined as studying the development or beginning of a particular disease. Okay, so again, pathology is studying disease in general. Etiology is studying the cause of a disease, such as the exact bacteria or the exact virus. And pathogenesis is studying the development or progression of that disease. The next two terms that you should be able to distinguish, even though you hear them all the time, you may currently kind of mix and match their meanings or use them interchangeably when they're actually quite different from each other. These two terms are infection and disease. Infection is the actual invasion or colonization of your body by the pathogens, so by the bacteria or the viruses or the fungi. Okay, so it's basically the pathogens taking over your body and being present. Disease then means the abnormal state that we observe with your body after it's been colonized or due to that colonization. Okay, so once again, infection means the invasion or colonization of your body by pathogens, whereas disease is the abnormal state the body ends up in due to that colonization. Okay, basically the actual feeling sick part. Okay. So you'll notice that since today's lesson involves a lot of terminology, I've added some slides for you to be able to write down or recap some of the information that I mentioned. Okay, I purposely did not put these in the PDFs that I gave you because, well, you have to actually listen to the lectures to get all of the information. Okay. So now this is to recap each of the terms that we just talked about. So you can pause the recording in order to write down all of these terms. Remember, terminology is very easy to be asked as multiple choice questions on an exam. Okay. Now, when we talk about diseases, infections, and all of this fun stuff, there is a reminder I want to make. Not all microbes are going to be causing infections. Not all microbes are pathogens. Okay? Remember that there is a lot of microbes on or in your body that are not doing you harm under normal circumstances. And what do we call those guys? Your normal flora. Okay, so what do we call the harmless ones in or on our body? The answer to that is your normal flora. Now, with all of those microbes in and on your body, there's another term that you sometimes hear, which is the microbiome, which is becoming a big topic in terms of research and publications. A lot of scientists are studying and understanding the microbiome for various purposes. What the microbiome actually means is all of the microbes, so that includes their DNA and their physical structures, as well as the environment that they're all interacting on. So for instance, one of the big things to study is the human microbiome. This is basically studying all of the microbes that are all over your body, as well as your body itself, because that's the environment that they're interacting with. Okay, we could gain a lot of information from that. And the last thing I want to point out on this slide is there are various things that can influence the microbiome, especially the human microbiome. So basically, the normal flora that you have on and in your body, they're always slightly changing, okay, from day to day, morning till night. Things such as your diet, your activity, were you very active? Were you sweating a lot and thus changing the pH of your skin? Did you go too long without eating? Or did you eat a bunch of salty things or a bunch of sugary things? Or did you drink a whole lot of alcohol? Hey, not judging here, because all of those things can affect whether or not the microbes want to be in that particular location and how well they survive. Also, even things such as your gender can affect your microbiome, okay? Because for instance, if you think about the various genders that exist, your bodies have different hormones excreted. So for instance, if we have a gender releasing a whole lot of testosterone, okay, that can affect the microbes that want to be present at a particular time. Okay, so there's a lot of different things that can affect your microbiome. Now, speaking of normal flora, this is something that we've mentioned quite a few times throughout the semester. We've been really focusing so far on the fact of normal flora being the good guys, all over your body, helping you out. But we do want to remind ourselves that even the good guys can be opportunistic pathogens. Okay, so what does it mean to be an opportunistic pathogen? Again, those are the bacteria or the microbes where normally they're being good, they're helping us. But if the opportunity presents itself, they will take advantage. Okay, so the two opportunities that I keep reminding you about are if you become immunocompromised, or if the microbe ends up in the wrong place. So for instance, E. coli that's supposed to be in your digestive tract, if it suddenly ends up in your genitourinary tract, boom, you have an infection and it becomes pathogenic. Okay, so that's what we mean by normal flora being opportunistic pathogens. Now, let's get back to the fact that they do normally help us and protect us. And the exact way that they do that is through microbial antagonism. Okay, so if you antagonize something, you're going against it, right? So normal flora can go against other microbes. Now, the way that they're protecting us through this antagonism, first of all, they're competitively inhibiting the growth of other pathogens, of other microbes. Because think about it, if the normal flora is taking up that space and using up those resources, there's not going to be any left, or not going to be much left, for pathogens to take advantage of, to flourish. Okay, so by simply just sitting on your skin, they're helping you out by taking up that space so that the bad guys won't sit there and start replicating. Some of them also produce bacteriocins, and that ending -sins is to show that they're toxins, but specifically to other bacteria. So bacteriocins are toxins that will kill bacteria And what the normal flora do is they will have toxins that kill the most closely related bacteria. Now, I know you might hear that and think, wow, what jerks. Why do they want to kill their relatives? Well, because think about it. The more closely related those other bacteria are, the more similar their needs are going to be, right? They're going to want the same resources, the same food, the same space. And we just mentioned competitive nature. Bacteria is not going to want that to happen. So that's why they tend to produce toxins that kill the more closely related bacteria, because those are their biggest competitors. In addition to that, just the fact of them, eating and excreting their metabolic waste products all affect the area that they're living in, which affects which other microbes would want to be there. So this last bullet point here is pointing out that the fact that these normal flora do things like affect pH, because think about it, I know you don't want to think about it, but they are technically excreting all over your body. So you got basically the bacteria version of urine and poop all over you. That will change the pH of that location. And as we've seen throughout this course, other bacteria, each little bacteria has its own preferred pH, own preferred kind of, environment that it wants. So by affecting pH and even things like the available oxygen, they're reducing the likelihood of other microbes being present. Now, this slide is a reminder of the 3 possible relationships that can exist between hosts and microbes. These are terms that I'm sure you've encountered in other courses throughout your educational career, but just a reminder, when we talk about relationships between organisms, okay? I know it sounds very romantic, but instead we're talking about symbiosis, okay? Symbiotic relationships are the interactions between 2 organisms. The 3 examples that we have here are commensalism, mutualism, and parasitism. So the easiest one that students tend to remember first is mutualism, because if something is mutual, both parties will benefit. So that's the example in the middle here. Mutualism is when both organisms interacting get a benefit, such as the E. coli living in our digestive tract. Okay, because they get tons of yummy food and good space to live in. And at the same time, their enzymes help us digest things that we wouldn't have been able to digest, such as cellulose of plant products. Okay, so you wouldn't be able to digest vegetables without their help. The next one that students find it easy to remember is parasitism over here. Because when you think of parasites, what do you think of? Well, I think of undergrads, but most people think of anything bad, right? Anything feeding off of you, taking your energy, taking your nutrients, just taking, and not really helping in any way, right? So in this case, parasitism means one of the organisms, so the parasite, benefits, but at the expense of the host, okay? So the host gets harmed or used up in this relationship. The example that we have here is H1N1. And if any of you remember H1N1 from the news quite a few years ago, H1N1 is swine flu. Okay, so any of the things like flus or colds, the things making you really sick, that'll be parasitism. Now the term that students tend to forget is commensalism. Okay, so we've discussed so far when one of the organisms benefits, when one of the organisms is harmed. So commensalism is when the host is unaffected. Okay, so you don't even realize that this microbe is present. It's not helping you, it's not hurting you. Okay, so that's commensalism, where the microbe will benefit, such as by getting food or space, whereas the host, completely unaffected, not helped, not harmed. Now, in this figure, they give the example of staph epidermidis on your skin, but I don't like that example Because if you think of any of the microbes living on your skin, what did we say about them? They're technically helping you out, right? That staph epidermidis is taking up space, taking up resources, so that the pathogens won't be able to live there. Okay, so I don't really like that example, but they use it because there are very few commensalism examples, okay, especially with regard to humans. You usually instead find commensalism as fungi living out in nature, okay, and we'll talk more about that in another lecture. The next concept to go over is etiology, which a few slides ago we defined as trying to study the cause of a disease. Now, there's a terminology that they use in this process, which is called Koch's postulates. So whenever you hear or see the word Koch's postulates, understand that this is a process to identify or pinpoint if a particular bacteria is causing a particular disease. Okay, so if you want to know for sure that this particular bacteria or virus causes this particular disease, then you would use Koch's postulates. So I listed them out on this slide, but in the next figure, we're going to get to see them visually. Now, when you look at this, there are four main aspects of Koch's postulates to prove or pinpoint the cause of a disease. The first one makes very good sense. The same pathogen has to be present in every case of the disease, because think about it. If someone has disease A, but doesn't have the pathogen that you're suspecting of causing disease A, well then how could that pathogen be the cause if someone has that disease without that pathogen present? Okay, so the first aspect of Koch's postulates is that the same pathogen must be present in every case. For the rest of these, I'm going to move on to the next slide so you can visualize them at the same time. So like I said, this is the visualization. After we know that the same pathogen is present in every case of the disease, then what we want to do here with Koch's postulates is you isolate or extract the suspected microbe from a diseased or dead animal, and you culture it. You then take that microbe and you inject it into a healthy animal and look to see, does that healthy animal then exhibit the same disease that we were studying? If they exhibit it, you then extract the microbe and study it to see, is that microbe the same one that you isolated from the initial dead or diseased organism? Okay, so again, what Koch's postulates is doing, you take a microbe you isolate from the diseased animal, you culture it and put it in a healthy animal, and if it is the true cause of that disease, it should then make the healthy animal show the same symptoms, the same disease as the initial animal. You can then extract it and study it to confirm that it even looks the same and is the same microbe. Okay, so once again, whenever you hear Koch's postulates, remember that the purpose is etiology, to pinpoint the exact cause of a disease, the exact microorganism causing that particular disease. Now, when we talk about these various infectious diseases, there's some terminology that you'll come across. This is another one of those slides where I say, I'm sure at one point or another, you've all used these terms or heard these terms, but you may not have known the true distinguishing aspects of the terms, or even if you were using them properly. So, the first term is symptoms. And your symptoms are the changes in your body function that are felt by a patient as a result of disease, meaning all of the things that you can visually see and feel due to having an infectious disease. Then we get to the next terms, and these terms are communicable, contagious, and non-communicable. Okay, all three describe infectious diseases, and you have to know the difference amongst them, as well as the examples of them. So first up, we have communicable diseases. Communicable diseases means that the disease can be spread from one host to another, but that it won't be that easily or quickly spread. So, for instance, the way to better understand this is to think of the examples. So the examples I like to give for communicable diseases are food poisoning and STDs. As you know, these can be caught from other people, but you won't catch it just by standing next to someone who's sick with one of these types of infectious diseases. Okay, so just by sitting next to someone who has an STD, you won't catch that STD. And as a New Yorker, I'm very grateful for that because traveling on public transportation, everyone would have an STD just by sitting and standing on those buses and subways. Now, when we then talk about contagious diseases, now it's a disease that's easily and rapidly spread from one host to another. So now with contagious diseases, just by sitting or standing in the same room as someone who has this infection, you can get it, okay? In fact, they might have been in that room a few minutes before you and left by the time you got there, and you can get it. So for instance, just sitting in a seat in a food court, okay, maybe someone who had the flu was sitting there right before you, sneezed all over that table, those chairs and all. It's all in the air, it's on the surfaces, and you can now catch it. Okay, so these are contagious diseases, things like the plague, like the flu, even like the common cold, where you can catch it very easily. And you notice with these, if someone in your family catches the flu, odds are you're going to catch it too, just by being in the same house as them. Okay, that is contagious. The last one is non-communicable diseases. Okay, so this disease cannot be spread from one host to another. So these are diseases you cannot catch from another person. Instead, you're catching them from things like soil. Okay, so the example of non-communicable diseases is tetanus. Tetanus, you can't catch tetanus from another person. Instead, you catch it from spores in soil. Okay, so from inanimate objects. Now, just a reminder, as I say the word tetanus, anytime you hear tetanus, I want you to think tetanus and botulism are both two very problematic infections because of exotoxins. Okay, very dangerous infections because of the neurotoxins that they produce, which are exotoxins. Okay. Just in case I spoke too quickly during the last slide, this is a recap slide so that you have a chance to pause the recording and write down what I wrote or what I said for each of the definitions that you need to be able to distinguish. And again, make sure you're always comfortable knowing the examples as well as the terminology. So now, like I said, this lecture is a whole lot of terminology, okay? And again, they're terms that I'm sure you've encountered before, but maybe didn't know the true usage of them, okay? So here we have terminology relating to controlling the spread of disease. So the terms we have are isolation, quarantine, and vector control. When we talk about isolation, I always have students circle or underline that I, that first I of isolation. So when you hear isolation, think I or Roman numeral 1, okay? Because in isolation, you are only separating the sick individual, just the sick person. Whereas in quarantine, you can circle or underline the first 3 letters, okay? Quarantine starts with qua, just like quantity, okay? Because a quarantine, you are now separating a quantity of people. You are separating all of the healthy individuals who had contact with the sick person and may now be infected, but in the incubation stage and not showing symptoms. Okay, so the example would be if you picture a classroom. If student A is sick and has an infection and we isolate or separate student A from the rest of the class, that would be isolation. What we would then do is take all of the other students in the class and separate them from the public just in case any of them have caught this infection, because you don't want them running around, going on the bus, going on the subway, going into stores or the gym or other classrooms, and now spreading this infection, which honestly, the concept of quarantine is very difficult to do. It's very difficult to control because, again, so many people don't realize when they are carrying around an infection before they start to show symptoms, okay, which we'll get to in the next page or the next slide. The last one here is vector control. What vector control is that you destroy the habitats or food or other resources that vectors rely on. And what we mean by vector are bugs, things like arthropods, things like mosquitoes. So the example of vector control that'll help you remember what we mean by this is the use of screens or netting. So think of mosquito netting and screens, or even all of the mosquito spraying that you saw a few years ago to reduce the risk of things like West Nile or Zika. So now, once again, I wrote a slide of some recap notes just in case you missed any of the information that I was saying on the last slide. So again, feel free to pause the video right now just so that you make sure to write down everything here on this slide. Now we have a very interesting figure for you to know. Okay, so normally you don't have to memorize any figures, but this figure you need to know each of these stages. This figure represents the development of a disease, the 5 stages of infection that you will go through no matter which infection you are suffering from. And yes, sometimes certain stages within this cycle will be less visual or less noticeable, maybe last a shorter time or a longer time. But no matter which infection you have, you will go through all of these stages, if you make it through that infection, that is. Okay? Now, when you go through this, I want you to not only be aware of the name of each stage, but also the key characteristics of that stage. So for instance, when we look at this graph, over here, when we see number of microbes, you can write severity, okay? So this axis represents the severity of your symptoms and of that infection, okay? So the higher we are, the worse the symptoms and infection will be at that point. Now, going stage by stage, the first stage of any infection is the incubation period that we were just talking about in the previous slides. This is the time when you have the pathogen in your body, but you don't realize it yet because there are no signs or symptoms. At this point, the bacteria or the virus is just settling in. It's going through those initial stages of the pathogen cycle, which we mentioned as attachment. So it's trying to really embed itself. And it's starting to replicate or prepare enzymes, okay? But it's not reproducing itself yet. You then head into the prodromal period, okay? Prodromal period is the second period of infection. What this means is you are now exhibiting the mild signs or symptoms. So what I want you to write for the prodromal stage or prodromal period mild general symptoms, okay? Whenever you see prodromal, I want you to think general symptoms. And if you think about any time you've just started to get sick, where you're like, oh, this is gonna be bad, I know it, it's usually when you get those general symptoms like malaise, okay? The really bad fatigue, or as I call it, that yucky feeling, right before you start to get really sick. So no matter which infection you have, during prodromal period, you'll have general symptoms that match any infection. So the big one being malaise or fatigue. The third stage in infection is the period of illness. Now, for this one, I want you to write specific symptoms. Okay, so in prodromal, you had general symptoms that you would get with any kind of illness. But for a period of illness now, you have specific symptoms. So for instance, if it's chickenpox that you have, now you'll get all of those red spots across your body. If it's an enteric infection, meaning your gastrointestinal, well, now the specific symptoms will be the vomiting or diarrhea. Okay, so now the period of illness, not only do I want you to remember that it's specific symptoms now, but I also want you to write down that this is when you may die. Okay, so if someone's going to die from an infection, it's during the period of illness. Then you hit the period of decline. The period of decline is, as it suggests, the symptoms are declining or decreasing. Okay, but I want you to put a star next to the period of decline and write that even though you start to feel better, this is when you can acquire secondary infection. Okay, so for period of decline, write down that you can acquire a secondary infection. Okay, why is that? Because think about it, at this point, now you have become immunocompromised, right? So if you're immunocompromised, what can happen? Opportunistic pathogens. So now that you're weakened, now your normal flora, who are usually the good guys, they can jump in and make you sick. Okay, you can also, because you're in a weakened immune state now, you can also pick up infections in the outside world where you may not have normally gotten sick from these things because, you were strong enough before to fight them before you really saw any illness. So that's why I always tell my students, be very careful when you're just getting over an infection. So even things like the cold or flu, because when you're starting to feel better, you're very, very susceptible to secondary infections. And a lot of times people will say, oh yeah, I relapsed because I was running around too much after I was getting better. It's not that you're relapsing from the same infection. It's that you're actually picking up a secondary infection. So please, anytime you're recovering from being sick, okay, even though you're starting to feel better, do not rush back to places like stores, the gym, or school, and even work, okay? Please try to avoid as many public places as you can while you are starting to feel better from an infection, because you very easily will pick up a secondary infection if you're not careful. And if you don't pick up a secondary infection, then hopefully you make it to the period of convalescence. And that simply means you're now all better, you beat it, and you've regained your strength and immunity pretty well by this point, okay? So now whenever you hear the term convalescence, just know that's a fancy way of saying you feel better. So if anyone asks you how you're doing, you say, I've convalesced, thankfully, okay? So again, make sure you're very comfortable with the 5 stages of infection, not only their name, but the significant aspect of them. So for instance, incubation, no signs or symptoms yet. Prodromal, general symptoms. Period of illness, specific symptoms, and you can die. Period of decline, feeling better, but can pick up secondary infection. And period of convalescence, you are back to normal, healthy and good. Ugh, I know, so much terminology in this lecture. Well, here's another slide full of terminology, and once again, these are all terms that I'm sure you've heard before, but we need to make sure that you're truly clear on how to distinguish them. So this slide, we're going to start looking at the duration and scope of disease, which means how long does a particular infection last, and how far does it spread? So when we talk about duration, there's a few different categories. An infection can be acute, chronic, subacute, or latent. Acute means any of the infections where the symptoms you'll notice will develop really quickly, but the disease itself will only last a very short time. So things like if you think about having a cold or the flu, they'll hit you very quickly, but they're not going to last very long and you'll start to feel better. Chronic, on the other hand, the symptoms you may not even notice at first are starting to build up. The symptoms develop very slowly in chronic illness and then last for a very long time. In fact, chronic illness can a lot of times even be lifelong. Okay, the big example of chronic illness would be things like TB or lupus. You then have infections that are subacute, and these are rarer to hear. Subacute infections means that they're basically an intermediate between acute and chronic. So with these infections, they'll develop slowly the way a chronic illness would, but they can kill very quickly the way an acute, very quick, short illness would. Okay, so with subacute, again, think of slowly developing, but then kill quickly once active, and a big example of this would be asthma. So for instance, asthma may progress very slowly, but if a child has a sudden asthma attack, it can kill them very quickly. The last one with regard to duration are the latent infections. So for instance, a lot of STDs go latent. Now latent, what that means is it's another word for dormant, okay, dormant or inactive. Now latent infections, what you notice with these is the causative agent, whatever microbe is causing this infection, goes inactive for quite a bit of time, but then will constantly reactivate and produce symptoms again. So big examples are things like herpes or shingles. Okay, and whenever you encounter or deal with any of the latent infections, the two things that tend to really make them reemerge or reactivate, one is if you end up immunocompromised, so kind of like when you have opportunistic pathogens as well. So being immunocompromised will reactivate latent infections, and times of stress. Okay, the more stressed out you are, the more likely these latent infections will pop up, which is why you'll notice that people who have, let's say, latent herpes or even mono, what'll happen is during times like finals week, suddenly you'll see the blisters kind of popping up and cold sores popping up. Okay? Now, in addition to duration, how long these symptoms are occurring, you also have the scope of infections. And what that means is how widespread on your body the infection is. So local is exactly what it sounds like. Local means the pathogen stays at the original location, so the infection is just at one spot. In fact, we usually call that spot the focus of infection. So for instance, if you get a splinter and you just have, that splinter in your finger, and your finger, that one spot is red, swelling up, painful. Okay, that would be a local infection. Whereas if that infection then spreads throughout your blood and your lymph and spreads to other organs, well, now that's systemic. Okay, so local is one spot. Systemic is traveling through your body. So blood and lymph is now included. There's some terminology with systemic infections that you have to recognize or be familiar with. The first one is septicemia. And what septicemia is that there are pathogens or microbes in the blood, whereas toxemia is specific to toxins in the blood. You then see two different forms of sepsis listed. And talking about systemic or sepsis infections is very important, especially if any of you plan on going into the medical field, or if any of you or your loved ones end up in the hospital, because I want to point out that over 250,000 Americans die each year from sepsis, including my own father. So I like to always point out and emphasize to all of the students, especially those of you going into the medical field, to be careful because systemic infections are becoming more and more common, especially in hospitals. Okay, and the two forms of sepsis that you see on this slide are severe sepsis versus acute sepsis. And when you see these terms, whenever you hear acute, based on what we were talking about a minute ago, acute means very quick. It all happens very quickly. So with acute septic shock, your patient can suddenly die within 24 to 48 hours because there's such widespread tissue and cell damage. Whereas with severe sepsis, they may have a couple of weeks before they're killed if you don't get to really culture and identify exactly what's causing that sepsis infection. Okay, so in severe sepsis, you still get the systemic inflammation and organ dysfunction, but it's at a lower rate, so there's still more time to try and save the patient. Where again, acute septic shock, within 24 to 48 hours, your patient can die. Okay, so make sure you're very comfortable with the differences amongst the various terms on the slide. And to help solidify your understanding of them, here's a recap of the notes again to write down, just in case I went too quickly over any of the terms we discussed. Okay, so once again, anytime you see a recap note slide, just simply pause the recording and make sure to write down all of the terms that you see. Now, the next thing to cover in this lecture is epidemiology. Whenever you hear epidemiology, that simply means the study of factors and mechanisms involved in the frequency and spread of illness. Okay, so basically we're looking into frequencies, rates, things like, how many cases of an infection are you seeing? Okay, there's a lot of terminology here you have to be comfortable with, and especially if you go in the medical field, you'll have meetings dedicated to epidemiology and looking at things like incidence, prevalence, morbidity, mortality. Okay, so our terminology, first one is incidence of a disease. So incidence would be the number of new cases that you're seeing pop up for a particular disease, and it's always given in a particular population and a particular period of time. So for instance, within 6 months in, Lakewood, we saw 200 new cases of whooping cough. And so we would say that the incidence of whooping cough in this location during that time would be 200. Now, if instead you are asked for a rate, then what you do is you put the number of cases over the total. So anytime you hear the term rate, then you should be giving a fraction as an answer. So in that same scenario for whooping cough, where I said 200 new cases in Lakewood during a 6-month period, the incidence would be 200. But the incidence rate would be that 200 over the population. So let's say we said the population was 100,000. Well, the incidence rate would be 200 over 100,000. The next terminology is prevalence. Okay, prevalence now, instead of just the new cases, this is the total number of people with that infection. So that means any of the new cases plus anyone who already had this infection. So for instance, if we said that those 200 new cases were on top of 500 already sick people with whooping cough, then the prevalence would be 200 plus the already 500 that had it. So that would be 700 as the prevalence. Okay, so the prevalence would be 700. And if instead I said prevalence rate, then once again you would simply put that 700 over the total population, which I had said in the example was 100,000 people. Okay? The next one is morbidity rate. And morbidity rate is simply another way of saying prevalence rate. Okay, so next to morbidity rate, you can write prevalence rate. Okay, because it means you're taking the total number of people with the disease, so that's new and existing, divided by the total population. Okay, so I'll never have you solve for it. You'll just have to write out the fraction. So for instance, morbidity rate in the example that I've been giving would have been the same answer that I said for prevalence rate. It would be 700 over 100,000, which was the total population. The last term to know is mortality rate, and please do not get the terms morbidity and mortality mixed up, okay, because that's a very common mistake, but they're two different things. Mortality rate is simply the number of deaths divided by total population. Okay, so if I said the mortality of the infection, it would be how many people died from it, whereas mortality rate is taking into account the total population. Now, when we talk about mortality rate, I want to point out that the total population never changes for that set time. Okay, you do not deduct the people who died. So, for instance, if I said that during that time that we were just talking about, 6 patients died from whooping cough in that population, we would say 6 over 100,000 is the mortality rate. Okay, notice we did not change the 100,000. Now, the best way to really understand these terms is to practice questions with them. So now on the next slide, I'll have an example, which is the exact format that you would find these questions on an exam or quiz. So here we have an example question, and like we've done in other lectures, you have to send me your answers to these questions in Remind by Saturday at noon. Okay? Now, what this question asks is it first gives you a scenario. It will give you a time period. It will give you information about a particular population. You then have to figure out things such as incidence rate, prevalence, and so forth. So I'll always first tell you a time period. So in this case, between January to June 2016, 110 new cases of whooping cough, pertussis, appeared in Tardisville with a population of 12,000. This brings the total number of citizens with whooping cough up to 212. Unfortunately, 5 people died from the disease during this time span. I want to point out that the other way that I can ask this, so instead of giving you the total number of citizens, I could have said that the 110 new cases were added to 102 people who already had the infection. So always read the question very carefully to see whether I tell you new and total, or whether I tell you new and existing, and you then have to add together the new and existing to find the total. Okay, so please make sure that you take a look at these 5 questions and send me the answers in Remind by Saturday noon as part of your participation in Remind grade for the total course. If you're having any difficulties, as always, just send me a message either through Remind or through email, and I will help you to guide through this question. The next thing you sometimes encounter when we're talking about things like epidemiology are the terms endemic, epidemic, pandemic, and also sporadic. Okay? If a disease is endemic, what that means is it's constantly present in a population. So the example I always give for that is malaria in Africa. Okay, so this disease is always present at a rather large amount. You then have, in this figure, sporadic. And as you notice, sporadic shows just every once in a while a little bit of a pop-up of new cases for a particular disease. So sporadic means only occasionally are you seeing new cases pop up of this infection. Then we have the terms epidemic and pandemic. And unfortunately, things like the news or the media have a tendency to mix and match these terms, even though they have two very different meanings. So if something is an epidemic, this means that the new cases of the disease are acquired by many people in a given area in a short time, meaning one location, one region suddenly has a large outbreak of new cases of a disease. Pandemic, on the other hand, if you notice here, this is now multiple regions. It is a worldwide epidemic. So for pandemic, what I usually do is I underline the P of pandemic, and I think of the singer Pitbull, Mr. Worldwide. So pandemic is when the new cases are popping up worldwide. And a big reason for pandemics nowadays, travel. Think about the airline industry. That opened up the whole world for infections to spread very easily, very far. Okay, so pandemics, a big part of it is thanks to travel. So now, once again, to recap, in case you had any difficulty writing down what I was saying, if I went a little too fast or anything, the 4 definitions that we had from the previous slide are sporadic, meaning only occasionally do you see that disease. Endemic, meaning the disease is constantly present. Epidemic, meaning suddenly in a short time in one location you have the disease popping up. And pandemic is when the epidemic has now gone worldwide. So once again, feel free to pause the recording in order to write all of these notes down. So speaking of pandemics and the spread of disease, when you think of the spread of disease, you can think either big worldwide spread or even within your own home, your own everyday life. Okay, there's various ways for disease to spread because there are various reservoirs of infection, meaning things housing the pathogens. So the first big category of reservoirs of infections are humans. Okay, and humans as reservoirs can either be sick people or they can be carriers. Okay, so what's the difference between sick and carriers? Well, are you seeing the symptoms, which is a sick person, or does it not even look like they're sick, but they are incubating that infection, which can still spread, and those are carriers. The other reservoir option is animals. And when an animal transmits pathogens, we call those zoonotic diseases. So think of the zoo, animals. Zoonotic diseases are when animals are carrying the pathogens. And you can get zoonotic diseases in more than one way. Okay, you can directly get them from things like, an animal bite, or you could get them transmitted from things like fecal matter. Okay. The next one are the non-living reservoirs, and these include things like water, soil, food, air, okay? And when you look at all the reservoirs on this slide, from humans to animals to non-living reservoirs, including water, soil, food, you have to ask yourself, which of these reservoirs would you consider the most dangerous? And out of all of these reservoirs, you may be surprised to know water is the most dangerous of all the reservoirs, because think about it, you cannot live without water. You can't find any alternative if the water is contaminated. Okay, humans, yeah, sure, you can avoid people. I do it all the time. You could avoid animals. I know some people who are terrified of things like dogs, and they will run across the street just to not walk anywhere near a dog. And non-living reservoirs, well, things like food. Okay, so on the news they say romaine has been, contaminated. Okay, I just won't eat a salad. Still plenty of other options to eat. Whereas water, you can't avoid water. Okay, so water we consider the most dangerous of the reservoirs. And contaminated water will cause infections like typhoid fever, cholera, okay, diarrhea, things that we talk about a lot in the lab when we do water control, quality control testing. Now, when you think of the spread of the disease from those reservoirs, there are various mechanisms that the infection can be transmitted. The first one is contact transmission. And when we talk about contact transmission, that means that you have either touch or close contact, okay? And contact transmission can be broken down into 3 different types of contact. There can be direct, indirect, or droplet contact transmission. So for direct contact transmission, that would be things like touching, kissing, biting, sex, any of those things, okay? Where you are directly contacting the sick individual, okay? Indirect, however, now you have a non-living intermediate, okay, to the sick individual. Now notice with indirect, okay, the non-living intermediate will be things that you associate with a sick individual. So for instance, needles, used tissues, hospital bedding, anything that you would have seen a sick person using, okay, that's indirect. Then you have droplet, and droplets would be if the sick individual sneezes, coughs, even laughs, and you get those little saliva airborne droplets. Now, when we talk about droplets, you should always ask yourself, what's more dangerous, smaller droplets or larger droplets? The answer is smaller droplets. They're more dangerous because if you think about it, if it's a small droplet, it could be airborne longer and further than a big, heavy droplet, okay? The next example of transmission is vehicle transmission. This is the one where pathogens now are hitching a ride on something you would think would be clean, okay? This would be something such as food or a fomite, like we talked about in lab. So in the case of vehicle transmission, the reason why it differs from indirect transmission, in vehicle transmission, the object is something that you don't associate with a sick person. Instead, it's an object, a fomite, that you would think is perfectly normal for anyone. Things like a doorknob, cell phone, glasses, your ring, your watch, okay, a shoe. Any of these things that it's not like you see a sick person coughing and sneezing all over it the way you would with a tissue or a medical needle, okay? The last type of transmission is vector transmission. And vector transmission can either be mechanical or biological. Either way, a vector is a carrier organism, usually an arthropod, or something such as a fly, a tick, a mosquito, any type of bug that you would think of. Okay? When we then categorize it as mechanical or biological, there are 2 ways that this bug can get you sick. In mechanical is physical movement, right? So mechanical vector transmission would be like if you see a fly land on your food and suddenly its body parts are contaminating your food. Okay, so it's brushing off with its little legs, looking like a little villain, rubbing its legs together. It might be brushing off pathogens onto your food that you then eat and get sick. So that would be mechanical when their body parts are contaminated and make you sick. Whereas biological, what's the other way an insect can make you sick? Well, by biting you. Their biological fluid. Okay, so mechanical is their body parts contaminating you, whereas biological transmission is biological fluid from a bite. So things like a flea bite causing the plague, or a mosquito bite causing Zika, West Nile, malaria. Any of those would be biological vector transmission. So again, just to recap notes, because there was a lot of information there, I wrote down the key points, and you can once again pause the video in order to write down all of the notes. Now, just to help you visualize what we were talking about, we're going to go through each of the transmissions in picture form. This first form is contact transmission, where the first one you see is direct contact, where you kiss or bite or have sexual contact with the sick person. Then you have indirect contact transmission, which is inanimate objects that the sick person has touched and that you associate with sickness. And then you have droplet transmission, which are the bodily fluids that are exposed through sneezing, coughing, things like laughing. Vehicle transmission, we have the examples of water, food, air, and also you may want to remind yourself fomites, okay, F-O-M-I-T-E, which are the inanimate objects such as a cell phone or a doorknob handle. Then we have mechanical and biological vector transmission. Again, the fly on food, that would be mechanical, whereas biting into your skin, that would be biological. Now, as we talk about the spread of infections, one of the biggest places where infections spread is actually the place you go to try and make yourself feel better after an infection. So it's actually the healthcare locations, such as hospitals or clinics or doctor's offices, where you actually tend to pick up the most infections. As we've mentioned before, we tend to call these nosocomial infections, meaning any infection picked up in a healthcare facility, okay, affects 1 in 25 patients. And as I said, the amount of sepsis in hospitals is jumping every year. And they result from things such as, the microbes just being present in a hospital environment, because that's where sick people go. Also, the fact that a lot of people there, since they're sick or dealing with recoveries, they're compromised. So we said immunocompromised patients end up being the most likely to pick up infections. And the last thing here is the idea of the chain of transmission, which in the medical field, we're constantly trying to break that chain. So the figure over here demonstrates an example of the chain of transmission in a hospital. You have the pathogens, which are currently in a reservoir, such as the doctor. That reservoir then has a portal of exit when he coughs into his hand. He then goes into a patient's room where he shakes the patient's hand, and that is direct contact transmission, without realizing that they have a cut on their fingers, and so you have a portal of entry, and the susceptible host, who is currently immunocompromised, quickly gets that infection from the doctor, okay? So when you're working in the medical field or when you're visiting loved ones, be very, very aware of the chain of transmission and try to avoid any unnecessary contact or any, how would we call these, maybe naive actions, okay? So basically meaning anything that you may not normally think of, like, oh yeah, my nose is a little, having some sniffles, let me wipe my nose with my hand. Well, not in the hospital, okay? So be very aware of your actions and always picture microbes and how they are getting spread. Here we kind of summarize some various ways that can help control these various infections, these hospital-acquired infections, okay? You want to reduce the number of pathogens as often as possible, so make sure to hand wash regularly. Use things like disinfecting the disinfectants on tubs or equipment. Okay, make sure to clean any medical instruments very, very carefully and very frequently. And when possible, try to use disposable items, okay? Because disposable items are kept sterile until you use them and then thrown away. So you reduce the risk of spreading any kind of microbes to other patients. Because sometimes, no matter how much we try to clean instruments, there are always going to be some highly resistant infections, okay? And also another thing that they try to do to help control these infections is by having control committees that really strictly regulate all of the actions within a medical facility. But again, your education and awareness are going to be the biggest things to control these infections. Be very aware of everything you're doing, and please be very careful, not only careful of the patients, but also be careful for yourselves and your loved ones, because you never want to track any of these pathogens you encounter back home to your loved ones. Now comes the fun part, which is diagnosing infections. And some of these ways, I know they're going to sound kind of complicated and not so fun if you don't have a strong background in things like DNA and RNA, but what we'll do is we will basically give a brief summary of each, okay? So we're not going to go crazy in terms of details and specifics. That's more for advanced micro, okay? Now, what I want you to be aware of are the 3 main ways of diagnosing infections, and be able to briefly explain each one, okay? How exactly can that diagnostic technique be used to show whether or not a patient has a particular infection, okay? So the 3 main ways we have listed here are growth analyses or biochemical testing, which is the first one. And you'll notice this first one, growth analyses and biochemical testing, is actually what we do in microbiology class lab, okay, when you work with things like selective or differential media. So basically, growth analyses and biochemical testing is looking to see how different organisms grow on different types of media to help narrow down or identify which infection is present. Serology is going to be the antigen antibody test that we'll look at in a minute. Okay, so whenever you hear serology, think antibody and antigens. And then the last one is my personal favorite, nucleic acid-based detection. And whenever you hear nucleic acids, think DNA. Okay, so we're going to go one by one through these. And again, as we go through these, make sure you're comfortable explaining some of the ways in which these can be used to diagnose a patient. Okay, so the first diagnostic category, as we said, is growth-based diagnostics. And this one is the easiest and cheapest one to do. As you saw, you're even able to do it in a college lab classroom setting rather quickly. In the growth-based diagnostics, you're basically dealing with the idea that microbes will have different characteristics when they're on different types of media based on their metabolic processes. So you have the idea of selective and differential media. So with selective media, again, that's the idea that on this media, the microbe will either grow or not grow. So certain strains will be inhibited, or certain species will be inhibited. And then you have the idea of differential, where depending on which organism is present, you'll have all different colors. So for instance, the idea of the MSA plate, the differential aspect is how you get the different colors depending on whether or not Staph aureus is present. So you see Staph aureus turns the media yellow. Okay. The other type of growth-based diagnostics that you see on this slide here is the enteropleury tube. And again, remember, the enteropleury tubes are only able to diagnose gram-negative strains. Okay? So depending on which organism you think is infecting the patient would determine which types of media you would use. There's also serology-based diagnostics, which will also be used in microbiology classroom lab. The example that we use in lab is ELISA, but there are various types of serology-based diagnostics. Now, whenever you hear serology, I always want you to think right away, antigens and antibodies. So remember that antigens are the proteins that are like name tags on the surface of pathogens. whereas antibodies are what the human host cell makes. Okay, the human host body will make antibodies specific to the pathogen's antigens. Okay, so now when we talk about serology-based diagnostics, the way you look at it is if a patient is infected with a pathogen, then that pathogen is in their body, so they now have that pathogen's antigens present in their body, and their body has made the specific antibodies to fight that pathogen. So you are basically looking to see, does their body samples, so their blood samples, their patient serums, their cultures, have pathogen antigens or even the antibodies against them. Either one will show you that the specific pathogen is present. The three examples that I want you to know for serology-based diagnostics are the slide agglutination test, Western blot, and ELISA, which ELISA is the one that you will be performing in this course, and it's called enzyme-linked immunosorbent assay. Now, the easiest one to understand or to explain, if you're ever asked to explain one of these tests, is the slide agglutination test. So what you see here is the slide agglutination test, and in this test, basically what happens is you place a certain amount of antigen, pathogen antigen, on this slide. So for instance, let's say you think that your patient has been infected with E. coli. You would put E. coli proteins on this slide. You would then apply the patient's serum on top of that and mix it together. What you then look for is, do they react together? Do they clump up? Okay, because what that would tell you is, if you have on this slide the bacterial or viral proteins, right, the antigens, and you apply the patient's serum, well, if that patient is infected with that particular pathogen, then their body has made the matching antibodies. And as you know, antibodies and antigens like to bind together. And that would lead to the clumping, which you see in this positive test result. Whereas if you have the antigen on the slide, and then you put the patient's serum on top of it, and that patient is not infected with that particular bacteria, then there's no antibodies present. There's nothing to bind and clump to that antigen. So it just stays a nice even smear. Okay? So with the slide agglutination test, again, if you had to explain it to someone, all you're doing is putting known bacteria antigens on the slide and then applying the patient's serum. If the patient's infected, they will have the matching antibodies and you see clumping. If they're not infected, there's no antibodies, so there's nothing to bind or clump together. And by the way, agglutinate or agglutination is a fancy way of saying clumping. So here you have the recap notes in case you didn't get to write down what I was saying with regard to the agglutination test. So please pause the recording and make sure to write down these notes to be able to explain what exactly is occurring in this serology-based diagnosis. Another example of serology is Western blot, and you see this a lot in research. So with Western blotting, it's a similar concept in that you're looking to see, does your patient have the antigens or antibodies that match up with this particular bacterial infection that you suspect. So in the case of Western blot, what you would do is you'd put all of your proteins from the patient onto a gel, okay? You would then have that gel transferred onto a thin membrane, a membrane that basically has pores to pick up all of the proteins from that gel. You would then block that membrane so that there's no what we call non-specific binding, and you'd add a primary antibody against the suspected pathogen's antigen, okay? Because here's the logic behind it. If your patient is infected with that particular bacteria, right, then in their protein sample, they'll also have that bacteria's proteins, right? Because they have that bacteria floating around in their body. So now, in their protein sample, you would have the bacterial antigen, and you apply a primary antibody that matches that antigen. So if they're infected, your primary antibody will stick to the membrane as well. If they're not infected, the primary antibody won't find anything to stick to and will get washed away. So now what you then add is a secondary antibody. And secondary antibodies are special in that they'll bind any antibody, but they also on their other side will have some sort of detection signal. So as you see in this one, the detection signal can either be color change or fluorescence. Okay, and so that will allow you to see whether or not your antibodies bound. Again, if they bound to this membrane, then that means your patient's infected and their body has the bacterial or viral protein antigen present. If they did not bind, then there's no matching antigens for that suspected infection. Okay. The last one for serology is ELISA, and that's the one that you'll perform in lab. This is a similar concept to Western blot, except now it's performed in wells or even in flat strips. Okay, now with this, what you do is you coat the bottom of each well with an antibody, so that you see here, an antibody matching the suspected antigen. You then add the patient's serum, and if they're infected, then they'll have the antigen that sticks to that antibody. You'll then apply another antibody that has detection on it, for instance, color changing detection. And if you see that binding and you get the color change, then you know that that patient is infected. Now, what I want you to remember for ELISA is the fact that pregnancy tests are actually an example of ELISA. Okay, so we'll see that in the next slide, what we mean by that. So over here, you see a pregnancy test to show you how the pregnancy test matches the concept of ELISA. And I always basically say that the pregnancy test ELISA is testing for the biggest parasitic infection that you can get that you're stuck with for life. Now with the pregnancy test, what you'll notice is it's testing for the presence in the patient sample of what's called HCG, which is a hormone that's only produced in high levels if someone's pregnant. So now what you do is you have this strip where one portion of it will have antibodies that are very specific to that HCG. Okay, they get pulled through the strip as the urine sample or whichever fluid sample is going through, and they bind to this area where the antibodies are specific to HCG. You then also have antibodies at the very end of the test that are general, that will bind any antibodies, including the ones that get pushed across the strip. This is just to show you a control of what the positive results should look like. Okay, so one more time, just to make sure this is all clear, if you look at it from this perspective, rather than on this diagram, you can see that the first portion of this ELISA strip will have a whole bunch of empty antibodies specific to whatever you're testing for. So in this figure, it's testing for this particular respiratory virus. In this area, the antibodies that would be present are specific to HCG. Okay? You then apply the patient sample, which will pull through capillary action these antibodies across the strip. If the patient is infected, then their sample will have the antigen or proteins that match the antibodies, get stuck to it, and they will then bind to the test strip area, where you'll see a color change. more of them will also pass through to the end portion, which is the general control antibodies that will bind the original antibodies no matter what, just so that you can see what a positive result would look like. If the person is not infected or not pregnant, then what happens is the antibodies from the beginning do not get any protein bound to them. No antigen binds them. So when they float through here through capillary action, they're not going to bind to the test area, okay, because there's no hormone, no antigen to bind here. They'll keep floating through, and then they'll bind to the control area because this control area is meant to stick to any antibody. So you get to see a positive result as a control to show what it looks like. And then you would compare and say, oh, my test area does not look like the control, so I am not pregnant. Okay? If this isn't completely clear right now, don't worry. In lab, we'll go through a little more details, and you'll get to really visualize exactly what's happening. Also, feel free to contact me if you're having difficulty understanding any of the diagnostic tests that we talked about. The last category is usually the trickiest, and I'm not going to go into the crazy details of it because you don't need that for this course. Again, we go into more detail in advanced micro. What I need you to understand is the idea that nucleic acids can be used for diagnostics. They will be the most accurate, but also one of the more expensive ways to diagnose the infection. There are a few different techniques that can be used that rely on DNA. The first one is PCR. If you take any genetics or molecular-based courses, you always get to run PCR, which stands for polymerase chain reaction. Now, what PCR is, in the case of diagnostics, is you'll have your clinical sample, okay? So, for instance, a patient's sample. Now, if your patient is infected with a particular bacteria or virus, you'll have the pathogen's DNA in addition to the patient's DNA in that sample, okay? So what you then do is denature that DNA sample into single strands. And what do you know about single strands of DNA or RNA? Singles are clingy and want to find their match, right? So if you denature their DNA sample so that it's all single-stranded, and then you apply these little probes, meaning these small single-stranded pieces of pathogen DNA that have little fluorescent detectors or some kind of detection on them. What then happens is, if the patient's infected, these probes will find their match in the sample, and they'll bind to it. You can then amplify them and get a detection. So for instance, you run them on a DNA gel, and you'll see the UV bands light up. Okay? What you mainly need to get from this is the fact that PCR, polymerase chain reaction, is an example of nucleic acid-based diagnostics. And also I want you to remember the fact that all of these nucleic acid diagnostic techniques are relying on the fact that single strands of DNA will try to bind to their complement. Okay, so that lets you probe for specific sequences. Another type of nucleic acid-based diagnostics that you'll hear about are things like hybridization or microarrays. Okay, so again, you don't have to know or understand all of the little details of this technique. Simply understand that what you do is you have the patient sample with the DNA release. You turn that DNA sample into single strands, and then you probe with single-stranded fragments of a sequence that matches the suspected pathogen's DNA. And those small sequences will have some sort of detection. So in this case, the small sequences, these little probes, have fluorescence. So if your patient has the particular pathogen's DNA, so in this case it's Salmonella DNA, this single-stranded little fluorescent probe will bind to your patient's sample of DNA because they're infected with Salmonella. You'll then see the fluorescence through that binding. If they are not infected, then the fragment will never find a match to bind to, and you won't see any fluorescence because they're usually made in a way that they only fluoresce when they're bound to their partner, and otherwise they get washed away throughout the technique. Okay, one more example of this, microarrays. So one more example of nucleic acid-based diagnostics are microarrays. It's the same concept that I was just talking about, just now it's on a specialized computer-looking chip, okay, kind of like you can see here. And again, it's that same idea where you will have these small fluorescent probes looking to see if the pathogen DNA is in the sample of patient's DNA. And if the patient is infected, then the pathogen DNA will be present, it will stick together with its match, and you will see fluorescence. Okay, so that's showing the fluorescence that you will see. Okay, if the pathogen is not present, if that suspected pathogen is not present, then the fluorescent probes get washed away during the process. Okay. So again, just to recap for what you need to know about nucleic acid as diagnostics, really focus on the examples, so know that PCR and microarray are examples of nucleic acids based diagnostics, and also understand that all of this is working because of the concept that single-stranded pieces of DNA like to find their match and stick together. Okay, so basically if a patient's infected, then their body will have the pathogen and the pathogen's DNA in it. And so if we denature the patient's DNA samples into single strands, we can see if pathogen DNA single strands will stick to that. And then we always have some form of detection such as fluorescence. Okay, so again, you can pause this slide and write down these notes just in case you ever have to explain what exactly nucleic acid-based diagnostics means. Okay, that is it for today. If you need any further clarifications, as always, please contact me in the Remind app. Thank you and have a great day.